Today, I will be discussing excitation-contraction coupling. Excitation-contraction coupling is the connection between the electrical action potential and the mechanical muscle contraction. A motor neuron is a nerve cell that originates in the spinal cord and synapse with muscle fibers to facilitate muscle contraction. Each skeletal muscle fiber is controlled by a motor neuron, which conducts signals from the brain or spinal cord to the muscle. Electrical signals called action potentials travel along the neuron's axon, connecting the individual muscle fiber at a neuromuscular junction. The action potential in a motor neuron triggers exocytosis of acetylcholine. The acetylcholine moves towards the ligand-gated channels. The ligand-gated channels bind acetylcholine and open. The net inward flux of sodium initiates an action potential. The action potential propagates over the cell membrane and depolarizes the T-tubules. Here, the depolarization approaches a voltage-sensitive dihydropyridine receptor. Also known as the DHPR, the dihydropyridine receptor has not modulated yet. Calcium is stored in the sarcoplasmic reticulum, both free and bound to calsequestrin. ATP-dependent calcium pumps are continuously active before, during, and after contraction. For each ATP hydrolyzed, 2 calcium are moved from the cytoplasm into the sarcoplasmic reticulum. Also seen here are the components of the muscle fiber. As you can see, the myosin heads are not yet in the cocked position. The depolarization reaches the DHPR and causes a conformational change that opens a ryanodine receptor calcium channel of the sarcoplasmic reticulum. Calcium diffuses out of the sarcoplasmic reticulum and into the cytoplasm. Calcium ions bind to troponin, and tropomyosin move to expose the myosin binding sites on actin. The myosin heads are now in the cocked position. Once the wave of depolarization ceases, the DHPRs return to their original conformation and the ryanodine calcium channels close. Acetylcholinesterase in the extracellular matrix of the synaptic cleft hydrolyzes acetylcholine to terminate the action potential. As the ATP-dependent calcium pumps decrease the concentration of calcium in the cytoplasm, calcium leaves troponin, tropomyosin blocks the myosin binding sites on actin, and the contraction ends.